Bismillah Rahman Rahim, June 24, paper 4-1. This is the third video on this paper. We will start with question number 7. It has been hypothesized that the mutation rate of an animal species may affect how fast animals of that species age and how long they live, lifespan. Table 7.1 compares the mutation rate and lifespan of 5 species of mammals. So mutation rate is per million base pairs per year. Species is cattle, ferret, horse, human, and house mouse. And mean lifespan. So 0.08 was the mutation lifespan. Mean lifespan was 20. Ferret was 0.20 and it was 8. Horse was 0.05 and 30. Human was 0.02 and the lifespan was 75. And house mouse was 0.28 and the lifespan was 3.6 years. So the mutation rate per million base pairs per year. Describe the relation shown in table 7.1. Now what is it showing you? As mutation rate increases, lifespan decreases. Mean lifespan 0.08 then 0.20 and the lifespan, mean lifespan in years was 8 years. So as mutation rate increases, mean lifespan decreases. So mutation rate increases, lifespan decreases. Then we come on to use table 7.1 to explain how these species may differ in the rate at which they can adapt to environmental selection pressures change. Higher mutation rate gives more genetic diversity. Short lifespan gives more generations per year. With higher mutation rate, shorter lifespan, they adapt or evolve better or more easily or faster. And mouse and ferret may adapt or evolve faster. Mouse and ferret, they are 0.20 and 0.28. And their lifespans are 8 and 3.6. So mouse and ferret, because they will have many generations per year, so they can adapt or evolve faster. So higher mutation rate gives more genetic diversity, shorter lifespan gives more generations per year. With higher mutation rate, they evolve faster. Mouse and ferret, having the shortest lifespan, will evolve the most. Part B1 of the question: compare the time scales needed for selective breeding and genetic engineering to increase the frequency of a rare allele in cattle, and give a reason for your answer. So comparison genetic engineering is very fast, selective breeding is very slow. And the reasoning is selective breeding occurs over many generations. So all cows could be surrogate mothers for engineered embryos. So comparison of selective breeding and genetic engineering increase the frequency of a rare allele in a cattle. So give a reason for your answer. There was only one reason was the comparison and another was the reason. So it will be C1 will be genetic engineering faster, selective breeding slower. So you had to compare and then you had to give a reason. So compare is C1 genetic engineering faster, or you could have said selective breeding slower. Reason is selective breeding occurs over many generations. Or you could have said all cows could be surrogate mothers for engineered embryos. Now coming to part 2, genetic engineering could be used to improve farmed animals such as dairy cattle. State 2 features of dairy cattle that could be improved by genetic engineering. Oh, what can we improve dairy cattle? Improve the quality of milk and improve the quantity of milk. So quantity and quality. So improve quantity of milk and improve the quality of milk, and that would get you your 2 marks for this part of the question. Now coming to question number 8, bears are several species of mammal in the family Ursidae. Climate change and loss of habitats due to human activity have reduced the size of some bear populations in recent years. The International Union for Conservation of Nature (IUCN) has a role in the conservation of species such as species of bear. Outline the role of IUCN in the conservation of species. So they will rank the species threat level or their conservation status. Are they really extinct, getting extinct, or are they before extinction? And then they will put them on the red list of the threatened species. And then they will influence or advise the governments or the countries who are the policy makers, and they will educate and bring awareness amongst the countries where they have to really improve the populations of the bears, and then promote a sustainable management of natural resources. Rank species threat level, red list of threatened species, then advise countries or governments or policy makers, educate and raise awareness, then promote a sustainable use of natural resources. Grizzly bears Ursus arctos horribilis and polar bears Ursus maritimus are found in North America. Figure 8.1 shows a grizzly bear and a polar bear. Grizzly bears have a varied diet that includes grasses, roots, berries, nuts, rodents, insects, and fish. A habitat of woodland or grassland, a maximum mass of 270 kilograms. Polar bears have a diet that consists of other animals such as seals. So this has got only animal, and these have got berries and roots and nuts as well. A habitat of land and sea ice, and a maximum mass of 450 kg. Wow, interesting. Climate change has caused population of grizzly bears to spread further north and population of polar bears to spread further south. In some areas there is habitat overlap, and on rare occasions breeding between grizzly bears and polar bears has occurred. The offspring of grizzly and polar bears are known as pizzly bears. Wow, I think it's a bit rude. Pizzly bears can breed together in the wild. Figure 8.2 shows a pizzly bear. With reference to three different species concept, discuss whether grizzly bears and polar bears should be classified as separate species. Grizzly bears and polar bears should be classified as separate species. Now, when it says with reference to three different species concepts, discuss whether grizzly and polar may be classified as separate. So, what was the species concept? Let's go back to that. Biological species concept said what? They're not separate species, they're the same species, as they can interbreed and produce fertile offsprings. Or you can say they're not reproductively isolated. So that was the biological species concept. Then the morphological means how do they look? Yes, they're separate species because they have different colors, mass, size, hump size. Then the third was ecological species concept. Yes, according to that, they would be separate species. Why? Because they have different habitats, they have different niche, they have different diets. And the fourth one was genetic or phylogenetic species concept. Now for that we are unsure because we have no information about the genetic similarity. So there were four concepts that we had to discuss, and that's why this is for 6 marks. One was the biological species concept, one was the morphological species concept, one was the ecological species concept, and the third was the genetic species concept. So I've given you the different headings which I've written in black. So the biological species concept, they are same because they can interbreed to produce fertile offspring. But morphological species, they are separate species as they have different colors. And the ecological species concept, they have separate species because they have different diets. Or they can even say they have different habitats, diets, or habitats. You could have said that as well. So ecologically, then genetic species concept, we are unsure because we have no information about the genetic similarity. Part C1, human activity has disturbed the habitat of many populations of spectacled bear, Tremarctos ornatus. The spectacled bear feeds on a variety of plant and animal species. Scientists compare the diversity of the food eaten by two populations of spectacled bear by analyzing their feces. One population lived in an undisturbed habitat, and the other portion lived in a habitat that had been disturbed by road building. The scientists analyzed the feces from 60 bears in each population. They identified the species present and the number of individuals of each species in the feces. The scientists calculated the diversity of species in the feces of the two populations using Simpson's index of diversity. The formula is this, and this is all given to you. Then it says table 8.1 shows the results and some of the steps for calculating the D value for the disturbed population total. Calculate the Simpsons index of diversity by completing table 8.1 in the spaces provided. Write the value for Simpsons on the dotted line. So we've got to write here, this is 3 marks. So how are you going to get the 3 marks? 1 mark for calculating the Simpsons index D. This one got you 1 mark. And then this, getting this line right, got you 1 mark. And then getting this line right got you 1 mark. So 3 marks. And this is how you got your 3 marks. Now it says part 2, the scientists calculated D value of 0.833 for the undisturbed population. Explain what is shown by the difference in the D values between the two populations of the bears. Undisturbed has a higher D, so has a larger or a higher diversity or a larger variation. See, what we analyzed was the feces. So what were they eating? So the D value for the undisturbed torch is 0.833. For this, it was 0.728. So undisturbed has a higher D value and so has a larger food diversity. What were we examining? We were examining what is present in the feces. So it means what do they eat? So this is how you have to answer this question. Now coming to question number 9, the harlequin ladybird, whatever, is an insect that shows discontinuous variation in color pattern and continuous variation in body size. So color show discontinuous in color pattern. So color pattern is either 2 or 3 types, or it's discontinuous. Variation in body size is very variable. So that is a continuous variation. Within a species, the variation that is observed for a particular characteristics can be described as discontinuous or continuous. Discontinuous variation has a different genetic basis from continuous variation. State 2 differences between discontinuous variation and continuous variation other than having a different genetic basis. So discontinuation has many phenotypes, or you can say distinct categories. Not a normal distribution, will only be a few bars on the bar chart, and no effect from the environment. Continuous variation is there's no distinct, there's many intermediates, and you get a normal bell-shaped curve and has a large effect from the environment. So continuous and discontinuous variation could have given me a whole lot of points, and we could have got away with the 2 marks. So whenever there is when you have to give differences and you have to compare of course means similarities and differences, but here it says state 2 differences, so between discontinuous and continuous variation. So I always say draw a line and write discontinuous on one side and continuous on the other side, and get away with doing a good comparison and getting your 2 marks. Figure 9.1 shows 3 of the color patterns seen in H. exeridis and the percentage of each of a population. So color pattern percentage, conspicua was 5, spectabalis was 13, and percentage 5%, 13%, and 82% suggest the genetic basis of the variation in color pattern in H. exeridis. So basically, why is there such a variation? Is 1 or 2 genes control the color pattern. Different alleles of a gene have a large effect, multiple or 3 alleles, and there could be gene interaction or epistasis. So you could have written any of these, any 2 out of these. 1 or /2 genes control the color pattern, then different alleles have a large effect, then multiple or 3 alleles are present, and gene interaction, or there is epistasis. Last part of the question, the body size of adult H. exeridis varies from 5 mm to 8 mm. Adult ladybirds do not grow or change size. Adults develop from larvae that hatch from eggs. State 2, environmental factors that may affect body size. Now I've written that already. Temperature or food supply, and during the larval development, and egg size or the diet of the mother. And now we come to the last question. Humans detect the sweet taste of sucrose sugar using chemoreceptor cells and taste buds on the tongue. The red admiral butterfly Vanessa atlanta detects sucrose using chemoreceptor cells located on its antenna and on its tarsi, which are the feet. The mode of action of the chemoreceptors in V. atalanta is similar to that in humans. Figure 10.1 shows the location of the chemoreceptor cells on the V. atalanta, and you can see the antenna are shown and the tarsi are shown, which are the feet. Describe how the presence of sucrose causes an action potential in the sensory neuron. That's a very important part of the syllabus. How the presence of sucrose causes an action potential in the sensory neuron associated with a chemoreceptor cell in the V atalanta. Now, we have lots of points here. Sucrose binds to receptor protein, second messenger, sodium entry, depolarizes, generator potential achieved, calcium ions enter, neurotransmitter vesicles move towards the presynaptic membrane, exocytosis in the cleft, binds to receptor on the postsynaptic membrane, sodium ions enter, causes depolarization, and this is only possible if the threshold of the action potential needs to be crossed, and that will result in an action potential, otherwise no action potential will be fired. So a lot of points here, and this was a 6-mark question. So we have about 10 points, and any 6 of these would get you your 6 out of 6. But either you know this or you don't know this. This was a very direct question. This wasn't something which was needed to be thought about.